Okay. Tomek, maybe you would like to start. Yeah? Okay, so I will take over your typical role. It's my pleasure to open next P4Health seminar, and it's my big pleasure to host today Andrii Domański from the University of Helsinki and Orion Pharma in Finland. Andrii is a neuroscientist with over 18 years of combined industrial and academic experience. He did a PhD at the University of Helsinki, and then he moved for the postdoctoral fellowship to the KFZ, Deutsches Krebsforschungszentrum in Heidelberg. And this is the place, in fact, where we met and spent together several years. Then, returning to Finland, Andrii started his own group in the Institute of Biotechnology at the University of Helsinki, and currently he is a senior research scientist at Orion Pharma in Turku, Finland. Right? Andrii, floor is yours.
Well, thank you very much for your kind words, for kind invitation to speak today. I feel very honored and privileged to meet you guys here and also people online, and also discuss during the next hour our views on preclinical development of drugs, new drug to treat neuropathic and musculoskeletal pain, which is part of Orion therapeutic areas. Well, so a few words about me. I spent most of my life in academia in University of Helsinki, I did my PhD there, and then I was working as a postdoc at the Krebsforschungszentrum, and the last 6 years I work as a senior research scientist in Orion Pharma. Most of my expertise is with neurodegeneration, and we did a lot of studies studying pathological protein aggregation in Parkinson's disease and ALS. But for the last 4 years, Orion decided to shift research focus and focus more on pain research, and this is what we are currently doing. So, well, probably everybody in this audience know what pain is. You've experienced, unless you have a specific genetic mutation which is making you like not feeling pain, it's actually very much serious mutation which would affect your health very profoundly. Most of people do, not only people, but also mammalian in some cases, even other animals. So most people do feel pain to different levels, but in general, this is a very basic and important feature for survival of the organism. And well, so official description of pain, which is scientifically accepted currently, is that it's like combining senses of both sensory and emotional experience. And it can be associated with actual tissue damage, but it's also associated with potential tissue damage. And so this, which makes it very, in fact, difficult to treat. And this emotional component is also very difficult to model in model systems, certainly almost impossible to do in vitro, but it's to some extent it's possible to model in animal in vivo situation. And if we look at the molecular mechanism or neuronal networks which are associated with pain processing. We see interaction of two different, of actually multiple different components. So pain arises at several different levels, and one of them being receptors, which are directly located in your body, in periphery. And these nociceptors, this is pain-sensing neurons, they transmit a signal through dorsal root ganglia to spinal cord, and then through spinal cord, vertical processing projection neurons projected to central nervous system. There are complicated networks involved in processing pain in the brain, involving several brain regions. As you see from this slide, this is pretty involving multiple neuronal systems. And then you have also this downstream pathway. So when you have both projecting neurons, like projecting from CNS down to spinal cord, and involved also in modulation of your sensory pain, modulation of your nociceptors, which is then also including this emotional component. Well, this is of course, a very simplified view, and what we are trying to do, we are trying to dissect these pathways on smaller fragments and try to model these interactions both in vitro by using neuronal cultures, but also in vivo using animal models. And for the next hour, I will explain you a little bit I will tell you a little bit about our efforts to model pain on these different levels, starting from basic functions of nociceptors and culture in vitro, and moving towards more CNS functions, and how can we study them. And most importantly, how can we apply those models to develop new drugs for pain treatments. So this is quite in a very simplified view, and I know that many people would complain that this is too oversimplified, but nevertheless, for the purpose of our talk today, let's split this pain into, let's think of pain of different types. So first of all, you have physiological pain. So if you cut your finger, or you expose yourself to a flame, or you expose yourself to some kind of dangerous situation, there are receptors, so there are neurons in your peripheral neurons, which express different ion channels usually, or different sensory proteins, which respond, getting activated by this impact, activated by heat, activated by cold, activated by mechanical forces, activated by chemical compounds like acids, and they are affecting electric conductance of sensory neurons, resulting in changing of activity of nociceptor, essentially, which is, as we believe, is directly related to the pain sensation, which is acutely affected. So there is this physiological pain. There is also types of pain which we call inflammatory pain, and in this case, this is driven by interaction between immune and sensory system, neuroimmune interactions, and there it involves more than one type of cells. In this case, we have also all these immune cells, which are releasing immunomodulators, which then interact with receptors on neurons, and again, affect cellular signaling components, changing the way how neurons are responding to the sensory stimulation, either activating in most cases, or actually sometimes also inhibiting neuronal activity, and resulting in this so-called inflammatory pain. When neurons start to degenerate, immune cells also play a role in this neuropathic pain component, where you start seeing pain based on processes which are activated during neuronal degeneration. This is also have some neuroimmune interaction, but this is actually more difficult to study. But also different cell types are involved in this part of pain. As I say, in real situation, in much more, our world is much more complicated than this. So probably for every particular type of pain, you have a mixture of all three components to different extent. Well, and the problems start when we get prolonged activation of nociceptors. So when you have, actually, we are pretty good in treatment of acute pain. And we are also pretty good in treatment of pain which is caused by acute inflammation. So NSAIDs like ibuprofen are pretty nicely working in most cases. However, in case of chronic pain, or in case of pain which is caused by more like central nervous system the problems, they are in many cases are not very efficient. And one of the things, again, this is a very oversimplified view, but one of the things of why pain is difficult to treat is that you have this, one of the options is that you have this peripheral sensitization and then also pain chronification. And well, for example, in case of this peripheral sensitization, it can be caused by multiple agents. Usually these are agents which are involved in immune reactions like cytokines, also immunomodulators, which are interacting with receptors on neuronal surface and by activating cellular pathways, including dysregulation of neuronal genes and changing the transcriptomic profile of these neurons. They're causing long-term changes in sensory neuron. It's called peripheral sensitization, and well, one of the effects of this peripheral sensitization it may be increasing expression of ion channels which are expressed on the cell surface of neurons, causing increased spontaneous activity of sensory neurons, or causing enhanced response to peripheral stimulators. We use it for assessing pain in rodents. We will get to that in the later on. So this can be measured, but it also can be measured on level of on molecular level. So basically, we can see changes in transcriptomic. We can see changes in proteomic profile of these nociceptors. Well, one of the reasons, one of the ways why we are doing this and how can we, if we are developing drugs to modulate pain, we need to, of course, develop the simplified systems in order for, at least for initial screening of the drugs, and also to understand molecular mechanisms affected by specific compounds, whether they can be used for modulation of nociception and activity of pain sensing neurons. So one possibility is to actually try to dissect the pathway down to very, very small components, in this case, sensory neurons, and try to develop in vitro platform where we can culture those sensory neurons in culture and follow their activity, spontaneous activity, or induced activity, which can be, for example, induced by applying immunomodulators, cytokines, to recapitulate, to see and model changes in neuronal activity, assuming that when we inhibit this neuronal activity, we would inhibit pain. In fact, this makes perfect sense because some of the known painkillers, some of the compounds which known to inhibit pain, they are working exactly like that. They are blocking activity of nociceptors. Well, for example, lidocaine is blocking activity. It's very nicely blocking activity of nociceptors and leading to complete numbness of innervated area. However, it has some serious side effects and then it cannot be used for general pain treatment. But nevertheless, so for us, in this level, we assume that neuronal activity is directly proportional to the pain we are experiencing. Very simplified version. And, well, it allows us for a relatively easy and cheap way to assess, initially assess activity of compounds on modulating neuronal activity activity of sensory neurons. Getting one step further, and this work we are trying to do, is to actually try, and when we achieve this goal of like having those nociceptors, we can get one step further, and we can make the system a bit more complicated. So we can add individual other components, other cells, making our system more complicated, getting some co-culturing sensory neurons with immune cells, so cells of immune system, and then trying to model neuroimmune interaction in vitro. How it looks on purpose? Well, so on a first level, we can prove that we can culture sensory neurons. We can dissect those neurons from rats and dissociate their dorsal root ganglia, dissociate their spinal cords, and move these cells to culture in vitro. We can also culture dorsal root ganglia organoids or spinal cord sections for several days or even weeks in culture. And we can model neuronal interactions and neuronal activity in vitro. Or we can start with primary rodent neurons, which are easy to get. But with development of iPSC technology, we can more and more frequently use human-induced pluripotent stem cells to differentiate them using different protocol, which are refined in every, like being refined all the time, to differentiate human stem cells into different types of sensory neurons and trying to study those functions of those sensory neurons in culture. So this actually pictures which are like done by our students, which we have, with whom we are collaborating with universities very actively, and also have programs for master students, so interaction between industry and academia, and at least in terms of education, we can also helping our colleagues to like mutually beneficial programs. And as a side note for some kind of academy-industry interaction. Well, so what can we do? So as I say, in this case, when we are trying to build in vitro pain model, and again, bear in mind that this is a very oversimplification. This is a huge oversimplification because we are, of course, missing these emotional components of pain and central components of pain. So it's rather in vitro model of nociception. So we can start with, as I said, with dissecting animals, and then we have those primary cultures. We can modulate those primary cultures. We can express or knock out or knock down different genes, either starting from transgenic animals or using viral-mediated transgenesis. We can transduce those neurons with genetic calcium sensors, for example. We can also measure activity of those nociceptors directly using multiple electrode arrays, or we can start with human-induced pluripotent stem cells. What is missing here, and which is very much wish, is working with primary human sensory neurons. This is a holy grail of nociceptor research. Unfortunately, they are very difficult to obtain, and in fact, so they cannot be obtained from healthy subjects, from healthy donors. So either it has to be done from deceased donors, or in some cases from cancer patients who undergo operations of removing their tumor. And in this case, sometimes also dorsal root ganglia are also unfortunately dissected, and these neurons can be then cultured. This has been done in several academic labs in the United States, and also some contract research organizations offer this type of studies, these types of models. However, it's still very, very difficult to obtain those primary neurons, and therefore we use human-induced pluripotent stem cells, which can be differentiated with some success to sensory neurons in culture. In our case, and this is what is done, this is what we did in-house, so in Orion. So we initially, as a proof of principle, we started working with rat dorsal root ganglia, dissecting them from animals, transducing with genetically encoded calcium indicators. And for non-neuroscientists here in the audience, can say that, well, you have these calcium levels as a proxy of neuronal activity. So every time neuron conducts its every time neuron gets activated, you see the changes in cellular calcium, which you can then follow by using calcium indicators which are fluorescent, and then by blinking this like activation of calcium, you see those activation, so like visualize neuronal activity in culture, as you can see here in this slide. And we started, because we are interested in neuroimmune interactions, we started with asking very basic questions of like, what will happen if we now stimulate these neurons with modulators of immune system, like cytokines, interleukin 1 beta being one of the example tool cytokines to stimulate those neurons. And our readout in this case is number of active objects, so the number of active neurons, as well as firing frequency of those neurons which is stimulated. In fact, you can measure this by following, as I say, calcium imaging. So transducing these neurons, for example, with genetically encoded calcium indicators, and measure fluorescence rate from these movies. We can also, of course, do more classical studies on following neuronal morphology. I think in this case you see development of axons after several days in culture for red primary neurons, but also we can use multiple electrode arrays, and nowadays even a new development is high density multiple electrode arrays to directly measure neuronal electric activity of individual neurons. And in this case, with high density electrode arrays, we can do it even with subcellular resolution, so we can, in some cases, even track the propagation of impulse, of electric impulse along the axons of these neurons. I unfortunately don't have these images here, but in very recent development of this HD-MEAs, it's possible to do, at least for some providers. This is kind of our dream to move it to the next level of precision. And with several of our proof of principle studies, which we are now hopefully publish very soon. So we see that depending on, this is a little bit complicated slide. So this is just a scheme of our experiment. So we put neurons in culture. After several days, we use again-associated viral vectors to express calcium sensors. By the way, even when we do this primary culture, they are not 100% neurons, of course. We also have other cell types coming from dorsal root ganglia of rat. And then we also can study this interaction between neurons and these other cell types as such. So what we see here is when we treat the neurons with interleukin-1 beta at different doses, we activate neurons, and this is now single dose of interleukin. We see activation of neurons in terms of burst activity, so they start firing more frequently with the higher dose of interleukin. And interestingly, so this activation lasts for quite some time. In this case, even up to 96 hours after addition of interleukin-1 beta. And when you get lidocaine, in this case, so lidocaine is a blocker of all sodium channels, so it essentially kills neuronal activity. So we see a very nice downregulation of neuronal activity when we apply lidocaine as a kind of negative control in these studies. We can also measure this activity of neurons on multiple electrode arrays. So as I mentioned, that you directly, so this allows you directly measuring electrical activity of neurons. And then again, what we start seeing, so when we induce when we add interleukin-1 beta to the system, the neurons which have been active before, they become more active. But also the neurons which are not active, so interleukin, not only it induces activity of existing neurons, which are like being active in culture, but it also induces activity of neurons which were not active previously, and which like general input, general activity, combined activity of these neurons is increased. So this is allowing us to also, so this system, in addition to seeing this interleukin-induced activity, this system actually allows us also to electrically stimulate neurons. And then we can also study how our compounds may affect basal properties of sensory neurons by seeing whether their response to electrical field stimulation is changed. We don't have this data yet in this presentation, but it's possible to do, and we are currently performing those studies as well. Of course, this system is also amenable transcriptome, so you can do proteomics studies on these cells. Also, you can do transcriptomics profiling of the cells. So this is a rather complicated slide, but my main point here is to say that so we actually, as a proof of principle, we did this to demonstrate that you do see in response to interleukin 1 beta stimulation, you do see upregulation of inflammatory genes and then downregulation of genes which are involved in neuronal function as well. So also demonstrating that in fact we can dissect molecular mechanisms of cellular mechanisms of our compound, affected by our compounds in relatively simple and controlled conditions. But so this is kind of, so far I've been telling you about activity of neurons. Yes, there is this neuroimmune interaction, and there is also interleukin 1 beta receptors on not only on neurons, but also on other cell types. And we do have other cell types in our cultures. But in fact, this is not, so to some extent we can do it, but in fact our system, as I mentioned, is pretty simple. And then the next question we asked ourselves is, can we actually make it a little bit more complicated? Can we adapt it to study interaction between different cell types, and one of the most straightforward choices was to study interaction of sensory neurons with cells of immune system. And in this case, we tried to do a co-culture of our primary sensory neurons with macrophages. Macrophages are known to be involved in modulating immune and pain response in osteoarthritis, or at least there is data from patients showing that macrophages are changing their type, also in osteoarthritis patients. And therefore, we've been interested if we can modulate this interaction in cultures. Indeed, and what I can show, again, is this a little bit slide which is quite complicated, but this essentially showing the same data, so this is quantification of this curve. So what we did, we cultured our sensory neurons, and then we added on top of these cultures, we added different amount of bone marrow macrophages derived from the same animal to avoid activation of immune cells from like different animals. So we did it from the same animal, which is possible to do. And what we see here, so when you treat the cells with lipopolysaccharide, which is activating immune response, in case of when we have only neurons, the response is rather small. But so response meaning activation of neuronal activity, stimulation of neuronal activity. However, when we have macrophages in culture, the neurons start responding much more actively with lipopolysaccharide treatment. They do respond to interleukin 1 beta in the same level, but with LPS we clearly see enhanced response of cells, of neurons being co-cultured with macrophages. And we actually demonstrated that depending on the amount of macrophages which you introduce into culture, you can see this difference in the response to LPS treatment. Meaning that, in fact, our system can be also used to study neuroimmune interaction and study drugs which are now affecting macrophages in order to understand how they would affect activity of sensory neurons. So, all things being said, as we know, this model still remains in vitro model. And we still, with all, even if we now, so the next steps would be to do iPSC cultures. We're also trying to do co-cultures and recreate the pathway in vitro with doing, you can also try to do sensory neurons with spinal cord neurons, and even in some cases, even CNS neurons. So there are publications showing that you can recreate pain pathway using different organoid systems, which is recapitulating those neuronal pathways. But nevertheless, this is our possibility to differentiate the cells, a possibility to modulate the cells is relatively limited. And like economic, we still need to use in vivo systems, so whole animal, in order to understand also CNS component of pain. And how could we do that? Of course, we are not the first ones to do pain in vivo modeling. So there has been already a lot of studies and models established in literature, in academic studies, and also in industrial settings with better or worse translational power to human. So you can treat rats or mice, in this case, with different modulators like inducing inflammatory pain. You can also induce neurodegeneration or like induce neuronal damage by physically damaging or restraining different types of neurons. Or you can also use different agents to modulate or model different diseases in the animal. In these few slides, I will be talking mostly about our model of, which is inflammatory pain model, which is induced by injections of complete Freund's adjuvant, inducing a component of multiple vaccines, which is inducing immune response. You can inject it either in the under the surface of the skin of hind paw of the animal, or you can also inject it to intra-articularly to the ankle, which is then induces huge inflammation, which lasts for a couple of days. You can, and it's also associated with pain, increased pain sensitivity. Or in case of osteoarthritis, we are injecting monosodium yogurt acetate. So this compound is actually killing chondrocytes, a cell type which is present in the knee and which with the time also leads to development of loss of the cells, development of osteoarthritis-like phenotype in the animals, and also induces pain in the knee. So this is a kind of model of osteoarthritis pain. You can also do surgical models, but we are not talking, we not touch them here, but this is something which we also actively trying to do in our experiments. Well, so we do all these manipulations of animal, but then, of course, animals, how you cannot really ask rats about how do they feel. And in fact, at least for mice, being a prey animal, apparently they evolutionary developed so that they are not really willing to show you the pain. Because in wild nature, if you are in pain, if you are showing that you are weak, you will be probably be the first one to be attacked and first one to be eaten. So mice are not very eager to show their pain phenotype, to show that they are being in pain. And it has been for quite some time in the big question in the field, so how do we actually assess pain behavior in rodents? And there have been several ways to do it, so just to give you a little bit of overview. So these are very classical ways how we can do it. We can do first and most straightforward is that we evoke pain by some kind of mechanical treatment. Well, probably, I don't know if it is now very popular, but certainly in my age, in my school days, it was pretty popular to put a pin on the seat of your neighbor and then let him or her sit on this pin, and then you hear the screaming and smiling for that. So we are doing, in fact, very similar situation with mice, where we can squeeze a knee or a paw of animal and well, of course, very gently until we see their response, until they become like restless, and then we measure the force which we apply in order to induce this pain response. And if the animal is sensitized, and now you remember my first of the slide about this peripheral sensitization. So if the animal has sensitized to a mechanical stimulus, the response comes with a lower force. And yeah, so similarly, well, you can also relate it to yourself. Is that you, for example, in summer you get a sunburn, and then when you touch your skin, when you have the sunburn, it's much more painful. The same touch gets much more painful when you have this sensitized nociceptors. You can also mechanically stimulate pores of the animals with calibrated force, calibrated filaments, which so-called this phone frame method, and the name of the person who was first described this. And then again, with a higher, so if the animal in pain, you induce response with lower force compared to control animal. The same goes for sensitivity to heat, and so you can stimulate with different variation of the method, but essentially measuring the same or very similar things, so response to heat or response to cold in this case. You can also then do more fancy studies when you try to present animal with a platform which have temperature gradient, so, and see where animal prefers to be in this case. So then it's like it's paying his animal, suggesting that if a mouse has more sensitive to pain, he will avoid areas of intense heat or intense cold. And then you can measure this behavioral as well. But in this case, we are measuring so-called evoked pain. So something which you stimulate and then you measure response. In many cases, when you talk to patients, they are actually suffering spontaneous pain. And this is an even bigger problem, or even more difficult to model in animals. Because the spontaneous pain, well, we kind of don't know when it can occur. If it is ongoing, that's easier. But if it is like pain which comes and goes in specific periods, it's quite difficult. So nevertheless, there are some ways to do this. We can follow the facial expression of rodent, or we can also start following stereotypical behaviorals like burrowing, and try to quantify the amount of burrowing gas, the animal gas in response, whether it's in pain or not in pain. In terms of osteoarthritis, when you have unilateral models, so usually we induce osteoarthritis, you induce injury of one side of animal, of one leg of animal, it leads to animal avoiding use of this injured leg and moving its weight more to one side compared to the other, which can be measured by measuring the essentially weight distribution between left and right legs of the animal. Or we can use so-called catwalk apparatus when we try to get animal like going and see how gait parameters are changed in response to pain, in response to induction of pain. Needless to say, is you can probably see and guess from this apparatus that animals need to be habituated to this type of measurement. This is not natural for rat to be confined to this type of apparatus. And in this case, the animals also need to be stimulated in order to walk. Sometimes they walk nicely, sometimes they walk not nicely, which represents a lot of experimental bias in the protocol, and more so for this facial expression, or in fact for this burrowing expression as well. So variability between individuals, even in wild type and non-induced pain, is pretty high. And it also depends very much on the person who is doing this study. So this is our own data. We see that both technical assistants, so in this case, it's mechanically stimulated pain using those foam-free filaments. And we have two research assistants, so one is relatively less experienced, the other one is relatively more experienced. The animals are the same, and depending on who is performing the study, you can detect or not detect the difference between the two cohorts. In this case, these are surgically induced osteoarthritis pain. So, trying to eliminate this experimental bias, we came with one of the recently published devices where we image animal from below. And it turns out that when we image animals from below, we see much more subtle changes in their behavior compared to when we image animal from above. So another advantage is that this is a so-called this black box device, is that it uses infrared camera. Also, in fact, animal is placed in the darkness, and so rats and mice, they are nocturnal animals. They feel safe in their completely dark environment. They think that nobody is observing them. And we can combine infrared images with also total internal reflection images, and which allows us to follow weight distribution between all different paws of the same rat. And you can also apply it both for mice and for rats. And then by using machine learning and computer image analysis, we can then, without any labeling of the animal as such, we can automatically extract positions of limbs and body posture, and then quantify this depending on whether now in our wild-type animals or in animals where pain has been induced. And so when we, for example, in this type of data, when we injected complete Freund adjuvant to one paw of the animal, we see the weight distribution is dramatically affected in that animal will avoid using injured leg. But what is most important, and this is of course not, so this is fully expected what we see, and this is very easy to, in fact, quantify also manually. But what is more important is that we can actually, by applying known painkiller, which we know that has effect also in humans. In this case, this is tanezumab. This is a monoclonal antibody which is binding to neural growth factor, which has been in the clinical development for showing efficacy in humans for osteoarthritis pain treatment. We see that by applying this tanezumab, we see that we can affect this pain response. We can rescue these animals, showing that not only we can see the phenotype, but we can also by using known painkiller affect this phenotype. And this is showing that, okay, well now if we apply our own compounds, we can discriminate between analgesic or non-analgesic effect of these compounds in this particular assay. Similarly, when we apply the monosodium urate, which is now osteoarthritis pain model, depending on the time in culture or after addition, we also see that we see nice development of this phenotype during time, which then can be rescued by tanezumab treatment. And this is now fully automated, and this is now can be done, and so these studies are in fact done by a master student in the lab, so you do not need to be highly trained, skilled research assistant in order to obtain this type of study, which is of course greatly simplifies our life as a pharmaceutical company. You can also analyze much more animals within a given time period, which allows us to get more robust, reproducible data, and also compare and cross-compare results from different studies and from different studies done in different research, in different projects, but even in different years. And so now, since like several years we've been doing this, we see that this is actually pretty much consistent data we obtain. We extended now to more neuropathic pain models. And in this case, this is a spared nerve injury. And so this is now still work in progress. But you can see, so for example, this rat has been suffering from this injury. And you see the weight distribution on normal, non-injured, and injured paw, which is very, very seldomly used. So this type of data you can now get with this newly developed methods of automated and unbiased machine pain assessment. And we believe that this is actually the future of behavioral analysis. Moreover, why we think so? Because now we are talking only about, so I've been showing you only like one parameter, weight distribution. But in fact, this system can be used to assess much more than weight distribution. It can also assess gait, so changes in the way animal moves, but it also can assess the other parameters like grooming, scratching, being on the like hind, it's like vertical movement, and this is currently which we are trying to also validate for pain, for assessment of pain, like in a more holistic approach. So we believe that well, it will also allow us to use more than one parameter for description of behavioral, so more holistic description of the whole animal behavior. And then you can do some kind of more complicated assays like multiparametric clustering. You start seeing the differences in behavior in response to compounds in different animal models and being more sensitive to assessment of subtle phenotypes, of subtle behavioral changes which you induce in these animals. All right, so well, for the next few minutes which are remaining, I would like also to talk about central components. So again, back to our, if you, if you still remember from what we started, original description of pain, also, so it has also the central and emotion, so like so-called emotional component, so central component, so brain is activated. And we also tried to see if we can model, if we can actually measure and image activation of brain areas responsible for pain sensing by building a kind of custom model with our university colleagues from University of Eastern Finland in Kuopio, where we used functional magnetic resonance imaged for rats in order to image their brain of rats in pain. So we used monosodium yogurt acetate model. So we had rats. We injected their MEA in their knees. And after 21 days, when we get to the so-called like chronic model of osteoarthritis, we imaged these animals in fMRI, in MRI scanner, but we also built a stimulation device. So the animals were stimulated even inside the magnet. They were stimulated with form-free filament for their injured paw. And in this way, we start seeing the response to pain. We start seeing the differences between stimulated and non-stimulated animals, but also between stimulated animals which have this pain-induced and the animals which were sham-operated animals. And we see the activation of several brain regions known to be involved in pain processing in response to the stimulation of like pulse stimulation within different time frames and different also at different times after mogul induction, and also we see the response depending on the strength of the stimulus. Moreover, if we now treat these animals with a painkiller, and in this case with pregabalin, we see that we can dampen the response, dampen this pain response in the brain, depending, again, so the same animals which now being in pain, treated with pregabalin, and their brain activity goes down. So we not necessarily claim that what we see is pain, but at least we believe that this is a little bit, so at least part of this brain activity is pain related. And we are, of course, now want to extend it also to the treatment with our own proprietary compounds in order so to more fully describe their action from in vitro model up to central stimulation. So, well, can we actually, are we now in the process of, so do we now have enough tools finally to enable preclinical drug development for pain? Well, we believe that at least we are much better and stronger position than before. So if you just want to remember something from this talk, so you can like have this slide. So we believe that we can model nociceptor activity in vitro, but it also with the help of modern ways of computer-based computer image analysis, we can start dissecting very subtle differences in pain behavior. And we can also start addressing more and more central actions in literally seeing the activation of brain regions related to pain in rodents. And this is very much in line with what has been done in human subjects. And so essentially trying to build a better preclinical translational models. And of course, our goal as a preclinical development is to build such models that would reliably and reproducibly predict human response to our components. And it's not that easy, but we think that with the help of current methods, we can do this. And as I say, so the next step would be using humanized animal models. This is something which is like really hotly studied, as well as induced pluripotent stem cells for modeling of human-induced pluripotent stem cells, derived sensory neurons for modeling human pain. With that, I would like to also to share my gratitude to my colleagues with whom we work in pain therapy unit in Orion Pharma, Ms. Miguel and Ilmarie and Lisa, who is also developing these machine learning algorithms, and Karina is our boss, and then we have our students, our research assistants, and of course our collaborators at BlackBox and University of Eastern Finland, Turku University, and all colleagues at Orion Pain Therapy Unit and University of Helsinki. And of course, thank you for your attention and for inviting me to talk here.
Thank you very much, Andrei, for this fantastic summary of the current methods. And now we have time for questions.
Hi, thank you for this interesting talk. Indeed, your company has a big portfolio of those tests. I'm just curious, how do you actually use it? Is it that you go from in vitro to in vivo? How do you decide which test? And because I understand you use it to test your drugs also. So what makes you choose certain tests, or do you do all of them? What's the pipeline?
Well, it's a very good question. Of course, we prefer to test initially in vitro. And, well, it depends on your target. It depends also on the molecular mechanism which you think your target is working. So, but certainly if we are targeting well, ion channels on the surface of neurons, this is something which can relatively straightforward can be accessed in vitro with neuronal cultures. And we, of course, try to avoid using animal models for ethical reasons if it is not necessary. So in this case, use of animals would come later on when we already have those tool compounds for then to of course study those tool compounds more advanced tool compounds. But in some cases, when our targets are non-neuronal. Yeah, well, we also, so that's why we're also trying to do also develop also this in vitro model for non-neuronal targets. This is more complicated because then you start seeing, you know, the more complicated in vitro system is, the more difficult it is to make reflect in vivo situations. So then we go in vivo as early as possible. Or sometimes we do in vivo studies for target confidence straight away. Well, depending on whether we have compounds to modulate target in vivo, of course, then we can go straight away to in vivo models. If not, then we can use genetic tools, both in vitro and in vivo. And so, but yeah, so in short, it depends on the target, but certainly from the point of like costs, we prefer to go first in vitro and do in vitro as much as possible, even in some very heterologous system like overexpress particular targeting hack cells and do some studies and then build from bottom to up. Yeah, so.
Yeah, I see, thank you. And I also thought when I saw this part about the central component where you use fMRI, so since you more or less know the pain regions, so what about using in vivo imaging techniques? Because you use calcium imaging in vitro, but you could also use it in vivo.
That would be very great thing to do, and we are certainly very much interested to do this. We have one practical reason is that we need, so in many cases you need to use this genetically encoded calcium indicators to do this in vivo, which means using transgenic animals. Unfortunately, in-house, we are not using transgenic animals, so we rely on our academic or contract research organization offering such studies. And it's not that many CROs which can do this. So we are actually very much interested in finding partners who can do this for us. So, yeah, I know that an academic partner is also in academia, this is great, and access to transgenic animals is much less restricted in academia than for industry. In fact, I was very surprised when I come to Orion and realized that, hey, I now have to pay for many things which, which used to come much cheaper. But yeah, so that's a great point. And in vivo imaging, both in rodents and possibly in dogs, this is something which would be great to have.
Okay, thank you. Victor?
This is related to the first question that you got, because you said that your pipeline would be go from in vitro and eventually the hits that you have in vitro, maybe test them in vivo. So that way you can actually test your in vitro models efficacy, at least you can look for false positives, right? Things that come up in the in vitro model that you think are good hits, and then the in vivo model actually shows that they're not working. You cannot see the opposite, but at least the false positives you can check. Have you done this? Is the in vitro, and have you used this data to upgrade your in vitro models?
Well, yes. So again, like a short answer, yes. So certainly this is one of the big advantages of doing this in-house for having both in vitro and in vivo under the same, well, in our case, in Orion. So you can, of course, outsource many of these studies, but then, exactly following your point, then you do not necessarily have the ownership of your model, so it's difficult to change the parameters, and you don't understand what's happening. So that's why we very much prefer, in the philosophy of the company, is to have at least small research and development activities in-house, exactly in order to do this. So now our expertise from in vivo models is helping us to refine our in vitro studies and vice versa. And
can't you just also use machine learning approaches on the in vitro models using the in vivo results to try and just optimize your analysis of the in vitro model without actually upgrading the model? So it would be easier for a machine learning model to find patterns that you think they're positive in the in vitro and they fail in the in vivo, and you could probably find them easily.
And this is a very good point. And so we hope that, well, now what we are doing, we have only few readouts. So now it's only like if you only follow calcium, well, you can do a little bit of machine learning when you follow this morphology. Then you get a lot of different parameters. When we see this for calcium response, we are lacking the amount of the data.
You're lacking resolution.
We are lacking resolution. So that's why I mentioned that with this high density multiple electrode arrays, this is something which we would like to get our hold on. And I know that many CROs are now starting to offer this service using those HD-MEAs exactly in order to get this resolution and also to get enable us to this multi-parametric analysis to extract more features from our in vitro systems. We are not there yet, at least we are not there yet. It does not mean that I know that some big pharmaceutical companies are doing this. I know that in the field of neurodegeneration, they are doing this already for quite some time, where they have much more complicated in vitro models than we do for pain, for those brain organoids and midbrain organoids. And again, my for neurodegeneration, I can give another 2 hours talk, but certainly this is something which we would be really very much willing to do, and this is a direction which we would be going. And the field is going there already, willingly, with or without us, so better with us than without.
Okay, thank you. And the very last question. I know that you cannot sell the company secrets, but if you could tell us what are the, let's say, current trends, drug classes in the drug development at the moment.
Well, of course, everybody was, so whole pain field was very much excited with a recent clinical approval of sodium channel blocker, specific sodium 1.8 sodium channel on sensory neurons, which Vertex has recently got approved, at least for acute pain treatment. But and to my understanding, field is more moving towards more neuroimmune studying also neuroimmune interaction, at least in terms of osteoarthritis pain, maybe less for neuropathic pain, but so it becomes more and more clear that we should look at other cell types in addition to neurons, and of course central modulation of pain. This is something which is still area of very hot studies. I mean, developing opioids without addiction potential, that would be very much. I don't know if it can be disentangled, but if so, that would be one of the things which would be very much interesting to do.
Thank you. Okay, thank you very much, Andrei, again. And now we will have a meeting for young investigators. So whoever of you is interested to talk to Andrei about his career track, you can stay and discuss.
Whoever feels young.
Not too many.